Hi, welcome to the BYU-Idaho Tutoring Center videos. Today we're going to be talking about complex inheritance patterns. So when Mendel was doing his research, he was fortunate in that everything he was studying mostly followed these more simple inheritance patterns. However, the world and nature is not that simple, and we see a lot of different more complex patterns that we have to try to explain and understand and factor in when we're dealing with Punnett squares and things like that. So first we need to differentiate between complete dominance and incomplete dominance, or the degrees of dominance that different traits can have. So in complete dominance, the phenotype of homozygous dominant individuals and heterozygous individuals is the same. So for an example, if we have two alleles, W, the phenotype, or what we see on the outside for the big W, little w, and big W, is going to be exactly the same. And then we come to incomplete dominance, and in complete dominance, the phenotype of the heterozygous individual is actually intermediate between the two homozygous individuals. So over here I have an example. If we have two alleles that determine the color of a flower, if little r results in a red flower, so that's the homozygous recessive, and if homozygous dominant results in a blue flower, then if we have a big R, little r, it's going to result in a purple flower. And so this is incomplete dominance because the heterozygous is kind of intermediate. So I know this isn't actually purple, but I didn't have a purple marker, so I just drew in black. And so this kind of shows that this one is not completely dominant over this one. And so the incomplete dominance might give credit to the idea that when we cross two things, we kind of get a mix of the two traits, which is what a lot of people believed about genetics prior to Mendel. However, if we do some genetics crosses, we're going to see that these dominant recessive patterns are still going to remain the same. So for example, if you were asked to cross a red flower and a blue flower with incomplete dominance, then you would know we had a little r, and a big R. Usually, if you were just asked to cross a blue flower and a red flower and it was following complete dominance, you would have a question asking, is the blue flower homozygous dominant or heterozygous, because either would give you a blue flower in complete dominance, and then we have to do things like test cross and stuff like that to figure it out. However, if it's incomplete dominance, then we already know that the blue flower is going to be homozygous dominant. So when we cross these, we're going to get all heterozygous individuals, and so that means that 100% is going to be purple flowers. However, if we then cross these individuals with each other, our big R, big R, big R, little r, what we get, my bad, big R, little r, little r, is not a mix, because if we're following that idea that we have kind of an intermediate between the two every time we mix them, then we would expect to just kind of get some more purple flowers, because that's kind of intermediate. But when we do the second cross, we notice that we have 25% blue, 25% red, and 50% purple. So that keeps the idea of Mendel's original ideas constant. And so then the last one is codominance. And in codominance, we have both alleles, or more than one allele, that are being expressed or exhibited. So a really common example of this is the ABO blood group in humans. If they have AB blood, or the alleles for A and B, then both of those are going to be expressed. So if you have A blood, then you just have this one red carbohydrate coming off you. That's how we're going to distinguish between it, this carbohydrate. If you have blue, then, or B, you're going to have this blue carbohydrate coming off you. If you have O, you're going to have none. But if we have AB blood, then we have both coming off. And so that's an example of codominance. Really fast, we need to make sure we understand how dominant and recessive alleles are actually physically being expressed. And so it's not that dominant alleles are somehow repressing the recessive alleles being expressed. More often, it's actually that the dominant allele is producing some enzyme and the recessive allele is producing some defective enzyme. And so as a result, if we just have the recessive allele being expressed with something like an RR, little r, then all we're producing is defective enzymes, and then something can occur and it results in some different phenotypic output. Whereas if we have at least one of the dominant alleles, then sometimes what can be happening is now we have at least some of our effective enzymes and they can do the job that the defective enzymes can't do. And so that's why sometimes these two phenotypes can result, and genotypes can result in the same phenotype. It's because we're at least creating one effective enzyme. However, in some cases, if we create both of these enzymes, then it results in some type of intermediate. So just make sure you understand that it's not that this guy is repressing this guy from even happening, it's more that they're producing two different things, and sometimes this one's defective, and that's why they produce different phenotypic outputs. And then something else is that a lot of times we think that the dominant allele is always dominant in a population, but oftentimes this isn't the case. A lot of times the recessive allele can be more prominent in a population. An example of this is polydactyl. If you have this, babies have this, then they will have more than the regular amount of fingers and toes. And so most people have just the regular amount of fingers and toes, 5 and 5. And so this is a dominant, and so what we normally have is recessive. So the majority of the population actually has the recessive allele for that. And then what can also happen is we can have multiple alleles. A lot of the cases we just say, oh, it's either the red or the blue, the big R or the little r, or the two only alleles we have. But a lot of cases we have more than one allele. Another example that we already looked at was the blood group. We have the IA allele, which is like the A blood allele, the red. We have the IB allele, we have the little I. So these two are going to be dominant to the little I. They're going to be codominant together, and they all just produce some sort of different phenotypic output. So all three alleles are factored in. And then pleiotropy, this occurs when one gene has multiple phenotypic effects. So we have, this is very common in diseases. Some of the hereditary diseases result in lots of different effects to the body that cause different problems. And so this is just an example. One gene codes for the flower color in pea plants and also the outside covering of the seed color in pea plants. And so one gene codes for multiple things. That's pleiotropy. And then epestis, this deals with a gene at one location influencing a gene at another. So what can happen is, for example, if we have one gene that codes for black or brown color in mice and another gene that codes for whether color is going to be there or not, if we say that we don't have color, then it's not even going to matter what this gene codes for, because it's not even going to be expressed if we say there's no color. So what's happening is gene 2, whether you have color or not, is influencing the color gene. And there's lots of different cases where this happens. If we have a gene that decides my hair color, but then we don't, and then there's another gene that decides whether I'm bald or not, or if I even have the ability to grow hair. If I don't even have the ability to grow hair, it's not even going to matter what that gene that codes for my hair color is going to be. So that's just another example where one thing affects another. And then polygenetic inheritance, it's kind of the reverse of pleiotropy. Instead of having one gene with multiple phenotypic effects, we have 2 or more genes that have one phenotypic effect. So kind of the reverse. And we see this a lot with things like quantitative characters. And those are just characters that are influenced, that aren't on an either or basis, they kind of have a range. So an example of that is height. We don't just have 2 heights that are determined just by 2 alleles. There's a range of short to tall, and it's a pretty big range. And the same with skin color. It's not just 2 skin colors that you can have, and everybody has either or. there's a huge range from light to dark skin color. And so we have multiple genes affecting one character, such as height. And so that gets a little bit more complex, but that's how a lot of the things we see in our bodies are determined. It's not just one gene determining my height, it's probably a lot more than one. And the last thing we have to look at is that the phenotype, or what we see, isn't always just based on the genotype. There's other things that can influence phenotype, and one of the main ones is environment. And so this is where multifactorial genetics kind of comes in. It's when we look at genetics and the environment to see how they influence the phenotype. And so there's a lot of things that can influence our phenotype. For example, there are some genes that code for my skin color. However, if I'm exposed to a lot of sun, then my skin color is going to change. That's how tanning happens. And so just because my genotype says that my phenotype should be one thing, my environment is actually changing my phenotype. And so there's a lot of different things that can influence phenotypes that are environmental, such as sun or pathogens or amount of exercise. And so another example is maybe you have the genotype to be a certain height, but if you are nutrients deprived or you're not getting the food and water you need for some reason, then you might not ever grow to be that height because you don't have the ability to. And so the last thing is just this applies to the norm of reaction, which is how much outside things can influence phenotype besides genotype. So for example, the blood group has a really low or almost non-existent norm of reaction. It doesn't really matter what outside things come in, you're still just going to have the same blood type. However, things like height and skin color, those have probably larger norm of reaction because outside things can affect them more. That's all for today. I hope you have a better understanding of more complex inheritance patterns. Just make sure you don't get incomplete dominance and codominance confused. That's a common one. So in codominance, both are expressed. So think of co, together, both are being expressed. And incomplete dominance, think of this word incomplete. The dominant allele is not completely over recessive, so we have this sort of blending of the two. And then also look at pleiotropy and polygenetic inheritance. Pleiotropy, one gene, multiple phenotypic effects. Polygenetic, two or more genes on one character. And so one way I remember this is poly means more than one, and genetic, so think genes. So polygenes, more than one gene effects. So just make sure you keep those straight. I hope this video was helpful. I hope you have a better understanding of these. You'll be required to apply these types of inheritance problems or patterns to different types of problems with Punnett squares and things like that. We did an example today, but I hope again that this makes sense. Thank you so much for watching and have a fantastic day.